So good morning, everyone, and thanks to the organizer for inviting me to present my work here today. So today I'm going to talk about how we can investigate brain interregional interaction from a lenses of dynamical system. And I want to do this in the framework of investigating how value forms in the brain. So how we associate different values to different external stimuli. So it is known that in the brain, the dopaminergic neurons broadcast information about unexpected rewards. In particular, for example, if we receive something to eat and we don't know if we're going to like it, we can smell it, we can look at it, but we still don't know, then we bite into it and we like it. And what will happen is that the dopaminergic units activate and spread all over in the brain information about the fact that we got an unexpected reward. The second time we get the same type of food, however, we already know we are going to like it. So the dopaminergic unit are going to activate directly, for example, as soon as we smell the food, and they will not activate when we eat it. So in this case, the dopaminergic units are conveying a reward prediction. So the prediction of a future reward associated to a stimuli. The dopaminergic units actually are conveying a reward prediction error, but I don't have time now here to go into this detail. So, and the dopaminergic units are convey this information and spread and release dopamine in a broad network in the brain, and they also have a glutamatergic co-release. And I want to study the formation of value in the specific network that involves VTA and the ventral striatum. The ventral striatum is important because it's known to be also highly involved in the formation of value. So the first thing we wanted to do is to investigate what is the sheer effect of a dopaminergic co-release together with the stimulus in the representation of the stimulus in the striatum. So what Leonard did, he recorded from the striatum while he presented to a mouse two odorant. One of the two odorant was paired with the release, artificial release of dopamine that he produced optogenetically, he triggered optogenetically, only for one of the two odorant. And what he observed is even after that he had this dopaminergic release, so when there was no manipulation, the response of the striatum to the specific stimulus, which was paired with dopaminergic release, was enhanced. So was stronger. So the dopamine induced plasticity in the network and reinforced the responses to the stimulus that was paired with dopaminergic release. Now, in Leonard's experiment, there was absolutely no reward. But what we wanted to know is that if this process is mimicking actually what happens when we get an actual reward. So Max did a similar experiment, but this time there were two others. And one of the two other, after a delay, was paired with a reward, actual consumption of reward. And what Max observed was indeed that the presence of a reward without the artificial dopaminergic release induced the same change in the responses of the striatum, which increased. Moreover, Max did experiment while recording in the ventral striatum and in the VTA at the same time. So VTA is where the neuron, the dopaminergic unit sits. And what Carla observed by doing an unsupervised detection of pattern of activity in this data was that there was a special type of pattern in the data, which was composed of a first activation of the striatal unit, this black one, and the later activation of the VTA dopaminergic units, the red one. And this activation, this specific pattern of activity, she found out that occurred only after the presentation of the stimulus, which was paired with reward. And also it wasn't there at the beginning. So it evolved with learning. So as soon as the animal managed to understand that that stimulus was actually associated with reward, we got this pattern of activity. So this whole observation led us to make an hypothesis on the formation of this reward predictive signal. So, and goes this way. So initially, we don't know that a certain stimulus is associated to the reward, so the dopaminergic units will activate when we actually get the reward. This would lead to a dopaminergic release in the ventral striatum, which will reinforce selectively the responses to the stimulus which caused the reward. And now that we have stronger striatal responses, these responses are strong enough to drive maybe the dopaminergic units, which at this point have to respond to the stimulus because the striatal units are responding to the stimulus. So from all this, we observe, we understand that there is a complex interaction between the striatum and the VTA, which it goes both ways. And it would be nice to have a quantification of this interaction and to model it. And of course, if we know everything about the system, we can do a biophysical model, and that would be the best option because we would actually understand completely the system. But unfortunately, this is not always the case. We can't always go to follow this path. So an alternative approach is not to attempt to build the model based on the anatomy of the system, but to try to build a model based on the function, do a functional model, which captures only the functional interaction between the element of the system. In the latest year in machine learning, there has been an incredible expansion of methodologies for reconstructing dynamical systems starting from the data. So the idea of these approaches is that what we observe, so any time series, but in this case, our neuronal recording, are the product of a hidden dynamical system that we can't access. This hidden dynamical system is captured by this latent space, which also receives information from the output like the animal does. So for example, if we receive an input that mimic the olfactory cue, an input which mimic the reward cue, and will evolve in time. This latent space, which is composed of different factors connected in a recurrent neural network, will then influence an observation layer, which is gonna be the one that we will ask to the algorithm to mimic the actual time series that we record. So to give you a concrete idea, here are the equation of the piecewise linear recurrent neural network, which is one of the type of this algorithm. I particularly like this type of algorithm among the other because it highly resembles what we are used to in theoretical neuroscience with the Hopfield model. So in this case, the recurrent neural network, the latent space is a recurrent neural network. And here are the equations. So the evolution of the latent space is given by an autoregressive term, an interaction term between the factor of the latent space and a constant, and then an input, a coupling with the external inputs. And here is all the evolution in time. And then we have how this latent space coupled with the observation model through this, to the observation that we see experimentally through this observation model. Yes. So what we need to do now with this very generic structure is to train the model to reproduce the data that we recorded experimentally. And with this procedure, we will eventually end up with a model. So it is shown theoretically that recurrent neural networks are able to fit to produce any dynamical system. So this tool is extremely powerful. And it can also predict dynamical systems that are not associated strictly to neuroscience. But since we are in neuroscience, we can also include some anatomical model. We don't have to for the quality of the model, but we can. And if we, for example, introduce the fact that we recorded this neuron in 2 different regions, then we can leverage this model to study interregional interaction. For example, if I force the loading factor matrix, this may be matrix which associate the observation model with the latent state model to be partitioned so that only some factor can affect only the recording of one region and the other factor can affect only the recording of the other region, then if I look at the equation of the latent space, what I obtain is the interaction matrix, which will tell me how much region A influences the future evolution of region A, and how much region B influences the future evolution of region A. This is a parameterization of the interregional interaction between these 2 brain regions. Okay, so we can apply this to the data we recorded. And for example, here you see the same session that I showed you before, the one in which there was this special activity pattern, this cell assembly, we call it in neuroscience. And you can see here in black and in red, the actual average trace of the neurons that we recorded, in black for the striatum and in red for the VTA. And in green, you can see the trajectories of the simulated trajectory by the model. So in this case, the trajectory are fully simulated. So I just give the very initial condition and I let the model evolve on itself. So it's not a fit, it's not a one-step ahead prediction, as many of these models usually show, but it's the full trajectory generated. And it's quite remarkable that it follows so well the stimuli. Of course, I had to give the input. So I have to tell, okay, now there is deodorant, now there is reward, and so on. So this is what we observe at the observation level. But we can also look at the latent space level, because the latent space is a more abstract representation of our dynamic. For example, if I build a model with 6 latent variables, 3 for the encoding of the first region and 3 for the encoding of the other region, I can now project the trajectory of one subspace, so one of the 2 regions, and see how it evolves in time. And specifically, for each point in time, I can quantify the influence in the activation, for example, of the units of the latent factor in region A, the influence coming from A and the influence coming from the other region. So I can see specifically where to the other region is pushing and if there is at all an input from the other region. I can also do something further, and I can try to play the neuroscientist and simulate experiment. So since I have now parameterized our interaction, I can also decide to block them in certain moment of the task. So for example, here I can select these 2 units, which are exactly these units which compose this pattern of activity that you see here. And what I can do is that you see here in green, the simulated trajectories for these 2 units when the network is intact. And in pink, you see the simulated trajectory when I actually block the projection of the striatum to the VTA when I present the stimulus. And what, for example, by doing this, because I have the W matrix, which I can block. So if I do this manipulation, what I observe is that in the VTA, there is no activation of the dopaminergic unit. So this functional model would suggest that the activation of the VTA unit at the stimulus paired with reward, so this reward prediction stimulus, is conditional to the activation of the striatal units. We can also test the opposite pattern of manipulation in which we block the projection of the VTA to the striatum. And what we observe is that actually this has the opposite effect and enhance the striatal responses. So this would almost look like that the later activation of the VTA is cutting down the response of the striatum to make it sharper. So to conclude, what I show you is that with this dynamical reconstruction algorithm, we can attempt to build functional model of our cognitive functions, which allow us to model them without any a priori information. These are good for many things. They have many applications. They can be used to study interregional interaction. They can be used to simulate experiments in our models. And something I haven't shown it here today, we can also ask more theoretical questions. So to which dynamical property different cognitive functions are associated and study the energy landscape, basin of attraction, and so on. From the point of view of value, what I show you is that dopaminergic release is sufficient to reinforce the striatal responses to stimuli. These reinforced responses are maybe driving the dopaminergic neurons to activate at stimulus presentation. And this dopaminergic activation cut down the striatal response. This, of course, required experiment because eventually that's the ultimate way to assess this. So I want to thank you all for your attention. I want to thank my collaborators, especially the experimental groups of the Kelch lab, so Max Scheller and Lennart Ottel and Carla Filosa, who did the first experiments, and then Daniel Dusewitz and Giorgia Koppe, which formulated the first version of the piecewise linear recurrent neural network. And I would like to also take the occasion to make advertisement because I just moved to Pisa, to Sant'Anna, and last week my lab has been approved, so there is no trace on the internet of this, but There is a brain dynamics laboratory, so if you're interested in neuroscience, physics, and machine learning, please contact me if you want a thesis, PhD, or collaborate. Thank you very much. I was super fast.
There are questions? So, to see if I understood, so in the broader picture of the interactions between sensory modalities and the substantia nigra, VTA, and striatum, I mean, at first it looks like, so the striking thing in the very first slide you showed us was a switch from reacting to taste, odor, glucose intake, whatever it is, the real reward, to the fact that you react to the visual presentation, okay? And so, I mean, I would like to understand how this fits to your conclusion, or how much your conclusions can help us to understand this. Is it, you're suggesting that the substance, the VTA, is actually activated by the first kind of stimuli, so the glucose, let's say glucose intake, while the striatum is stimulated by the visual stimulus, and then tells the VTA you should activate as if something good is now here. Is it what you say?
Yeah, so there are many pathways in this whole network. And of course, this initial response of the VTA is not driven by the striatum. So it's driven by other regions. Okay. But the region of the striatum that we selectively recorded is the olfactory tuberculum, which is the region where there is the direct input of the olfactory stimuli to the striatum. And what they observed both in vitro and now with the Leonard experiment in vivo is that the co-release of dopamine, it helps to increase the button, the synaptic button from between the olfactory input to the striatal SPN neurons. This plasticity then allows the next time that the same stimulus come to selectively